Hello, students. Today, the topic is the search for genetic material. The search for genetic material. And we know that Gregor John Mendel has proposed principles of inheritance. Gregor John Mendel has proposed principles of inheritance in the year 1865. And he stated that the characters from parents will be in the transferred to the children in the form of factors. And in the year 1869, Frederick Miescher discovered the nucleic acids. Okay, in the year 1869, Frederick Miescher discovered the nucleic acids. So, both these events occurred or took place almost in the same time. But the DNA is not proved as genetic material at that time, and it took very long period to prove the DNA as genetic material. And the most important experiments which have proved DNA as genetic material are these three. One is transformation experiments conducted by Frederick Griffith in the year 1928. Okay, and he said that the genetic material could be a transforming principle. And this transforming principle is later characterized, but that is what biochemical characterization of this transforming principle was done in the year 1944 by three scientists. And you have to memorize their names. One is Oswald Avery, second is Colin MacLeod, and third is Macklin McCarthy. Okay, Oswald Avery, Colin MacLeod, and Macklin McCarthy. And we can say that the final proof or the final evidence as DNA as genetic material has come from this experiment, the third one, evidence from experiments with bacteriophage, which are in shortcut are called Hershey and Chase experiments in the year 1952. I hope now you are understanding in which year the DNA is confirmed as the genetic material is in 1952, means what actually they were discovered in the year 1869. Long duration was, long gap was there. Okay, so these experiments, evidence from experiments with bacteriophage were conducted by Alfred Hershey and Martha Chase. And in this session, we are going to discuss the first one, the transformation, okay, experiments conducted by Frederick Griffith. So now, the first one, transforming principle. And the experiments are called transformation experiments. And here, Frederick Griffith has taken this bacterium Streptococcus pneumoniae, and he recognized two strains here. One is S3 and the other one is R2. And this S stands for smooth, is what smooth walled, because it contains what a capsule or a slime layer or a mucus layer is present. And R2 is rough cells, and here that capsule or mucus layer is absent in these bacteria. And what he has observed is that, okay, let us see. And he has used the mice as experimental tools here. So, when he injected S3 cells into mice, mice got pneumonia and they died. Mice died. That indicates that these S3 cells are virulent. Means what they cause? Disease. And later, when he injected R2 cells into the mice were alive. So that indicates that the rough cells are avirulent. Now, what he did is that he killed these smooth cells just by heating, and heat killed S cells. Heat killed S cells, S3 cells. And these heat killed S3 cells, when they were injected into the mice, nothing happened to the mice. Means they were alive. So we can see that R2 cells and heat killed S3 cells are not harmful to the mice. And later, what he did is that he made a mixture, and he mixed R2 cells plus heat killed S3 cells. Of course, here already we have come to know that both of them are harmless. So, when this mixture is injected into the mice got pneumonia and they died. So, they died. And now he has done post-mortem for these dead mice, and he recovered. He should recover what cells? You see, R2 cells, but he recovered, to his surprise, okay, living S3 cells. Okay, he recovered living S3 cells, and you can see that there is no, actually there are no S3 cells because these are heat killed S3, and he should get R2 cells. Instead, he got what? S3 cells, living S3 cells. From this, he concluded that these S3 cells are none other than these R2 cells only. R2 cells, they have received some substance from heat killed S3 cells, and they have changed into S3 cells, and he called this as what? Transformation. And the substance which they acquired from these heat killed S3 cells is called transforming principle. Okay, and so in the next session we will discuss how this transforming principle was identified later. In the next session we will see that.